Now, I don't know if anyone has seen this picture of the Pope in a white puffer jacket, but it came out this year in May or March. And the day it came out, I started receiving it from a bunch of people. And of course, now we know that it's not real, it's computer generated because we're talking about this subject. But at the time, people didn't know that. So it really begs the question, if the computer generated image looks so real that the person cannot distinguish it from the real Pope, does it really matter that it's computer generated? And I don't have an answer for that. And generative modeling has such a broad range of applications. So there's this website called This X Does Not Exist. And you can replace X with a lot of words. And there is a generative model for that. So this chemical does not exist, for example. It gives you a 3D render of a chemical and with the correct bond pairings. So they claim to have solved the problem of drug discovery because you can just reload the page over and over again and test those chemicals and see if you cure cancer. And there's also the sneaker does not exist, which generates images of sneakers that could have been made by Nike or Adidas or any mainstream brand. So today we're going to talk about generative modeling. What are the goals? What can we do with it? And try to formally define it. Then we're going to talk about the taxonomy of these models, try to find GANs in the family of different models, talk about the pros and cons of each, before moving on to GANs and how we can improve them to make it work with different kind of data modalities. And then we'll talk about conditional GANs, which will be a stepping stone towards arriving at causal GANs, which we will need to understand the talk in next hour about GRouNdGAN. All right, so generative modeling as goal, usually we are given some training samples from some probability distribution, and we want to learn a model that approximates that distribution really well. And once we have perfected it, there are two tasks that we can generally do with these models. The first one that we have on the right is density estimation. So with density estimation, we have a collection of samples, examples, and that collection can be very large. And we're trying to learn the underlying density function that represents that data. And this really is at the core of a lot of unsupervised learning tasks, because since we don't have any labels for these problems, we really have to find the underlying structures and patterns in the data. So that's why density estimation is quite a useful task. The second one is sample generation. So with sample generation, you can see over here, we have training samples from some probability distribution P data. In this case, we have faces of humans. And we want to learn a model that learns some P model of X that approximates the training probability distribution. And if that approximation is very accurate, we will be able to generate samples that are very close to the ones in our training set. Now, you might ask, wouldn't it be redundant to generate samples from a distribution from which we already have some samples? Because we need some training samples to train our models anyway. And I would say yes and no. No, because in a lot of fields, like in single-cell RNA-seq, we have very rare cell types, and we don't have enough examples for those cell types. So if we are simulating data, we can use this computer-generated cells alongside real ones to improve the performance of a lot of our downstream tasks, like classification, pseudo-time analysis, etc. Second thing that generative modeling can solve is filling in missing values. So once again, in the context of single cell, if you think of dropout events as being these missing values because of low mRNA abundance, most methods that exist try to borrow information from neighboring cell types, neighboring cells, to make some statistical assumptions about what is a dropout probability, for example, from a cell. And that introduces some bias, and rarer cell types, these models don't really generalize to those. So if we're not generating some augmented data set, we can use that as reference instead. And that is what SCI-GAN does, for example. And in another area is semi-supervised learning, when we don't really need labeled data. So we can generate so much unlabeled data with generative models and, again, improve the performance of these tasks. Other applications are generating time series data for planning possible futures or training reinforcement learning agent in simulated environments. And as we talked before, with density estimation, it's really important to understand the latent representations of our data, and these can be used as features in a lot of downstream tasks. Now, if we look at this very nice tree that classifies generative models into different families, and it comes from a tutorial from Ian Goodfellow, the first place that this family tree splits is when we ask ourselves, does this generative model explicitly model density with an explicit density function or not? If the answer to that is yes, we'll move to the left side. And again, we ask a second question, is that density function tractable? If yes, we are at this bottom left side of the page. And defining some function that is tractable for such a high dimensional distributions that are very complex is difficult. But there are methods that are able to do so. And the main advantage is that they can directly optimize likelihood of training data. And that likelihood function can be used as a means of evaluating these models. But the con is that they're very time consuming when generating data, especially with pixel RNN and CNN. So now if we give up on having a tractable density, we will arrive at a class of methods similar to variational autoencoders, where we have a latent vector Z. And because of that, the function is no longer tractable. Instead, we're doing some approximation and optimizing a lower bound on likelihood. And the main con of VAEs is that it's been shown in different publications that usually they generate lower quality samples compared to GANs. So now, if we completely give up on this left side and not try to explicitly model density, we will arrive at GANs. So with GANs, we are not necessarily interested in modeling density, but we want to be able to generate samples from some probability distribution without necessarily knowing the underlying density. Now, the problem with doing that is that we can't just directly sample from such highly dimensional complex distributions. So generate, for example, images, audio, or cells. So the main idea of GANs is to say, okay, what can we use to sample from? And as simple as it gets. So the simplest distribution from which we can sample is a Gaussian distribution or random noise. And then we try to learn some mapping, some transformation from that random noise to our distribution, which is our target distribution. It can be images in the example we saw before, cells, etc. So we pass in some input noise Z to some function which we represent with a neural network, and that function will try to learn the data distribution of our training set. And once trained, this function will hopefully be an accurate transformation from this latent space to our target space. So the generator we saw before, that is not trained. So how do we train this so it will actually work? The high-level idea is that we take a game theoretic approach and we define two players. The first player we saw previously is called the generator. And the goal of the generator is to generate samples that are very realistic. And we call those fake samples because they're not real. What is real? And the second player is a discriminator. And as the name suggests, it takes the samples from the generator that are fakes, along with samples from the real data set, and then tries to distinguish between them. So it is discriminating between the two. So the idea here is that these two agents, these two players will co-evolve in a symbiotic manner. And the Nash equilibrium of this game, our global optimum is that the generator is so good, it always generates realistic samples, and it always confuses the discriminator, and the discriminator has an accuracy of 0.5. Now, how do we train these two agents? Because we can't train them at the same time, right? And because we can't train them at the same time, we first train the discriminator in one iteration and then train the generator in the next one. The way discriminator training works is that we pass some input noise to the generator that generates samples, which are fakes. And along with some real samples, we pass those into the discriminator. So the discriminator doesn't know which ones are real, which ones are fake. However, we know that. So we can calculate some binary cross entropy loss, setting label 1 to be real samples, label 0 to be fake. And this loss is used to update the parameters of the discriminator. So if we now look at the objective function at the bottom, the discriminator wants to maximize its objective so that d of x, that is, its predictions on real data, are close to 1, and dg of z, its predictions on fake data, is close to 0. So we do gradient ascent on the discriminator. Now, when training the generator, we now freeze the discriminator, or when we're training the discriminator, we freeze the generator, and once again generate some fake samples, pass them into the discriminator, which will find some kind of cost. Here we calculated again with binary cross entropy, setting labels to be 1, because the generator wants to confuse the discriminator, so it wants those to be detected as 1. And here, the generator's objective is to minimize dg of z so that it's close to 0. So that means the discriminator will make a prediction close to 1. So the discriminator will make predictions and predict that those samples are real. Now, putting these two costs together, we get this value function at the bottom of the page where the generator is trying to maximize this and the discriminator is trying to minimize it. And these two losses are at odds with each other. So that's why we call them generative adversarial networks. They're adversarial and adversaries. And now training is very similar to any other deep learning method. We just choose an optimizer. Very common choice is ADAM or AMS grad. And we simultaneously generate a training mini batch and a fake mini batch coming from the generator. And we follow the process that we saw. And a lot of people find it useful to train the discriminator for more times than the generator. So usually K in this case, for every 1 time we train the generator, we train the discriminator for 5 times, and that is seen to help with stability issues. So now that we have trained our GAN, we can generate new samples. In this example, we are generating realistic images of faces. So we can choose 1 point in this noise dimension, which we call a z vector. And that noise will be passed into the generator, which will map it to a point in the target distribution. So this point over here generates this face of a young man with hair. Now, if we take a different point in latent space, the generator will map it to a different point in target space. So now we're generating a picture of a man with a receding hairline and glasses. Now, what we can do is to control what we are generating. So we can do latent space interpolation between those two z vectors and interpolate in the target space. So the example we saw is the second row of this picture, and we see that the young man is being slowly transformed into the other man who, so his hairline is starting to recede and he's getting glasses on. And you can even do more complicated kind of latent space arithmetics. So if we take a z vector that generates the man with glasses on the bottom here, and then from that subtract the z vector of a man without glasses and add the z vector of a woman, we will get an embedding that passed into the generator will generate now a woman with glasses. And why is this important? Because we're not able to do so in pixel space itself. If we try to do such arithmetic operations, we will just get some superimposition of a bunch of images, and that is not meaningful. Now, you can already see how we can apply this to transcriptomic data, right? Say that you have 2 cells, one is differentiated fully and one is undifferentiated. This first paper that I believe it's the first paper that came applying GANs to single cell, they tried doing this interpolation. So taking those 2 cells, finding their z vectors, and then interpolating in that latent space and generating cells along the differentiation path. And when now they compare the results in terms of how some genes are upregulated or downregulated following that differentiation path, they find very similar results to a pseudo-time inference method like monopole. But anyone who's worked with GANs will tell you that they're really hard to train, especially if you try to follow this vanilla formulation, which we just introduced. And a very common issue of non-convergence is what we call mode collapse. And mode collapse happens when the generator generates only a subset of the data set. So in this case, we have handwritten digits from MNIST. So the generator generates a batch and it is able to generate from all the digits. However, the discriminator finds that most of these are fake, so it labels them correctly. Now, when the generator receives feedback on the next round, it will only generate 3s and 8s, which weren't correctly identified by the discriminator. And on the next run, it will only generate 8s. So here we see an example of mode collapse because it's not generating from all the different digits. Now, why does this happen in a more formal, let's formalize this. And that's because the minimax game is not the same as the maximum game in GANs. And we are trying to solve for the minimax game, not the maximum. If you're doing maximum game, it is as if you're setting the discriminator constant and only updating the generator. So the generator has no incentive to generate new samples from classes that the discriminator is doing really well on already. So this is another example we have, and you see that the target samples are on the target modes are on the right. So we have 8 modes, and at every 5,000 steps of training, the generator collapses to a different mode. And that's because at some point, the discriminator will realize and correct itself and updates. And there are many solutions that have been introduced to solve this problem, like unrolled GANs, but the most well-known one, the very well-respected one, is using a Wasserstein distance. Now, why do we want to use the Wasserstein distance, and how is it different? So if we now look at this left figure, we have a generated distribution in blue and a red distribution in red that correspond to generated and training distribution. So the discriminator wants to push these two far away, and the generator wants to bring them together. And we see the difference between the left figure and the right figure. The left one is obviously better for the generator. However, when we're using this cost function, which we studied, it's very similar to Jensen-Shannon divergence. And because there's no overlap between these two distributions, we will get a cost that is pretty much identical between these two cases, although the left one is better for our case. Now, what the Wasserstein distance does is that it doesn't only rely on this probability dense distributions. Instead, it tries to estimate the cost that it would take to take the generated distribution and then try to mold that into the shape and space of the training distribution. So that is as if we're taking each of these blue points and then transporting them into the red distribution. So Wasserstein loss will give us a different cost for the left case and the right case. And what this means is that we have very meaningful gradients during training, even when the generator is doing really bad. And now, because the discriminator with Wasserstein loss is no longer giving us values of 0 or 1, it's not discriminating. So we call it a critic because it outputs real values. Now, there's one condition that we have to enforce on the critic when we're using Wasserstein distance, and that is one Lipschitz continuity. And it means that the norm of the gradient has to be always less than 1 everywhere. So we enforce that using gradient penalty by penalizing the critic if it has too large gradients. And we add that to the objective function. Now, if we take this improved version of GANs and apply it to single cell data, we still get really bad results. So this is from the paper that does that exactly. It is training a Wasserstein GAN with gradient penalty, applying it to single cell data. And here we have Tesney plot of real versus fake samples. So the generated ones are in red and real ones are in blue. And we see that although there is some overlap, these generated space don't cover the entire variation within a cell subpopulation. So a later paper that came out called SCGAN, they tried solving that problem. And the way they do this is by integrating some domain knowledge, which we have already. And that is using library size. So a very common pre-processing step in single cell is to library size normalize the cells to some number like 10,000, 20,000. And what SCGAN does now is to actually enforce that condition at the output of the generator. So generators' output are always normalized to library size. And because we're now giving this extra information to the GAN, we converge way faster with way less stability issues compared to before. And you see here in this new Tesney plot of real versus fake, we're covering the entire variation within subpopulations. Now, an extension to Weierstein GANs and just GANs in general is causal GAN. And causal GAN basically means that you can control from which population you're sampling from. So if you have different cell types, you can condition on a cell type and only generate cells of that specific cell type. And that's done by passing the labels as a one-hot encoded vector to the discriminator and the generator. And that's the simplest way of conditioning. Of course, there's like more complicated ways that are marginally better than this. Now, when SCGAN tried to introduce conditioning, they were successful at doing so. So we see over here that they're generating real and fake cells that are kind of overlapping for one of the clusters. And they show that they can now use this conditional GAN to generate from rarer cell types that they have downsampled and improve the performance of downstream tasks like classification and trajectory inference. And that wasn't very good on that rare cell type with very few samples. Now, we talked about conditional GANs, about regular GANs, and we're arriving at what we're anticipating, which is causal GANs. But before that, we have to understand the difference between conditioning and intervening. So take this population that we have in our training set, and for simplicity, we only have male and female. So male in blue and female in orange. Now, when we condition on a specific label like mustache, and we set that to false, a generative model will only output females of the population and some of the males. And that's because we have very few female samples that have mustaches in our training set. And on the other hand, if we condition on mustache being true, the conditional GAN will only output men because it's more likely that men have mustaches. Now, what is conditioning doing here is only limiting what is generating to a specific subset of our data. When we're intervening, we are forcing all of the population to have a mustache. We're forcing them to grow mustache. And we represent that intervention with a do operator. And we can also intervene on no one having any mustache, so setting label mustache as being false. And that will force everyone to not have a mustache. Now, this might seem like a very silly example with mustaches, but you can easily draw the parallel between this and, say, randomized control trials, because if you're just observing the population and some people have taken a treatment, some people haven't, that is what we call conditioning. If now you're no longer just observing the population, but forcing them to half the population has to take this treatment, the other half has to just not take this treatment, we will arrive at intervening on that population. So we are controlling what that population is doing. And then how do we really represent this? We can think of causal generation as a conditional GAN task, because a conditional GAN is technically causally generating an image or any sample based on a set of labels. The only problem is that it's not capturing the dependence between these labels. So here in our example, we had labels male, which causally controls mustache, and then in turn, mustache generates an image. So when we're doing this intervention, it's as if we are removing any links between mustache, the label on which we're intervening, and its ancestor, and then setting that to be a constant, like true in this case. And this will only influence the downstream and the descendants nodes, which in our case was the image. So let's see more concrete examples of what we just saw. So as we saw, conditioning only limits the generated data to a specific subset of the data. So on this left figure, we see what happens when a conditional GAN generates some images of people's faces based on a label mustache equal to true. We only get males in our population because they're more likely to have mustaches. So we're simply observing the data very passively and seeing which ones are true. Now, when we're intervening on mustache equal to 1 following what we do on the causal graph, which we just saw, we're, in addition to generating from the male population, generating females with mustaches. We're forcing them to grow mustaches. And that's why here we're generating from an interventional distribution and not from an observational one. And having an interventional sample can be very useful because it's very difficult to estimate causal effects when you only have observational data. And you can apply this, as I said, to who has taken a treatment, forcing people to take a treatment, or forcing some cell to have some kind of perturbations, for example. Another example of that is conditioning on bald versus intervening on bald. We have more bald men in our training sample. So conditioning on bald will only return us pictures of male who are bald. But now intervening on this label bald will also give us some female examples who are bald. So we're forcing them to become bald, basically. Now, what's the problem with all the GANs that we previously saw with conditional GAN, with normal GANs? It's that they can only sample from these observational distributions. And that means that they cannot sample from distributions that they haven't seen before. So they cannot sample from out of the distribution places. So they cannot sample females with mustache, for example. And that is because they can't capture this dependency that we have between the labels on which we are conditioning our image. So now the idea of causal GAN is that given a known graph, so we know what is the causal graph generating some data. Here we have, for example, age controls gray hair, which in turn generates the image, or age causes people to have eyeglasses, and then that generates an image. We want to now impose this into our GAN architectures. And doing that is very difficult because we can't at the same time train on these labels and also generate some images. So the authors of causal GAN took an approach where they tried to split this task into two. The first task is everything, all the labels before the image. So they first asked, okay, how can we capture these causal effects in a generative model and then train a generative model over those labels? Once we have those labels generated by this causal model, we can then condition the generator of the image, which is the sync node of this graph, based on those labels. Now, how can we create this first part, and integrate the causal graph? It is way simpler than you might think. So take this example where we have X and Y together generate Z. That is as if we have three functions, f of X, f of Y, and f of Z, and each of them are getting some inputs. So f of X and f of Y generate X and Y labels based on some noise, similar to what we saw before, which is just a generator. And then Z receives X, Y, and also some noise, and then generates the label Z. So we're basically having a bunch of generators together, and these generators inherit the structure of the graph which we want to impose. So once we have created this generator which follows the causal graph, we can simply train that generator in a Wasserstein GAN framework similar to before. And that will generate all the labels X, Y, Z for us. And in the examples we saw, those labels were, for example, mustache, gender, age, gray hair, etc. So once we have this first part trained, this generator, we rename it to something called the causal controller, just so we don't confuse the name with the second GAN, which we're going to train. So we take the generator of that first GAN, which generates us labels based on that causal graph. So it captures the causal dependence and causal effects between the labels. And we pass those labels into a generator along with noise, similar to, actually pretty much identical to a conditional GAN that conditions on concatenation. And that generator generates us an image which should capture the labels that are passed in and by the causal controller. Now, the problem we face in here is that we don't just want to generate an image, but we also want that image to be generated following the labels that are passed in. And if we just train that in a GAN framework, the generator will not capture those labels. So we have to force the generator to capture these labels L of G, which are passed in by the causal controller. The way we do this is by adding two different networks. One is called the labeler, the other is the anti-labeler. And they both try to estimate the label that generates an image given the image. So you pass in an image of, say, a man with a mustache and glasses. It is trying to estimate a mustache equal to 1, eyeglasses equal to 1. And then we can train this GAN with any framework we have in mind. In the paper, they trained it with a DC GAN architecture and also with a BGAN architecture. So in the next talk, we'll see how we can apply this to single cell data and what are the challenges we face and how can generating from interventional distributions be extremely helpful in the context of computational biology.
I'm sure you're all aware of gene regulatory networks. In general, these are network depictions of relationship between genes and the regulators, for example, transcription factors. And in general, these are complicated networks. But a first order approximation of this that a lot of times we can use is this bipartite graph that I'm showing here on the right, where we have transcription factors regulating genes. And this simple model has shown to be useful in describing many different gene expression programs in different contexts. Now, the idea of gene regulatory networks has been quite exciting. And for years, scientists have been interested in inferring them in different conditions, in different contexts. And originally starting from, for example, bulk transcriptomic data, and nowadays with single-cell RNA-seq, as well as other data modalities, various GRN inference methods have been introduced. Now, one of the problems that we typically face when we want to develop a GRN inference method is that how do we tell if we are actually finding the correct GRN that we are supposed to find? And this is not unique to this GRN inference problem. In general, when we are talking about network reconstruction, this is a very challenging task. So how do we deal with that situation? Well, traditionally, different approaches have been taken. For example, one common approach is to look into the literature, try to use, for example, text mining approaches or manually curate data sets, or rely on databases that have curated evidence from many different experiments. For example, String has protein-protein interaction information. KEGG is a famous pathway collection, or GTRD that, for example, mainly collects TF-specific ChIP-seq data in order to give us kind of a core gene regulatory network. Now, of course, when we are integrating data from many different sources, from many different contexts, from many different biological systems, there are issues. First of all, we end up with a network that may be prone to errors, may have edges that are not reproducible, or you may have situations where you have missing edges in your data. But maybe more importantly, we know that these GRNs are context-specific, they're tissue-specific, they're cell type-specific. But when we are relying on a single database, in most cases, you're aggregating over many different conditions in order to have statistical power, which means that your ground truth end up being not specific to the biological system that you may be interested in studying, and you may have, for example, single-cell RNA-seq data to study it. So what is the ideal scenario? The ideal scenario would be to use, for example, perturbation experiments or TF ChIP-seq data for that particular data set or that particular biological system that you want to study. Now, these technologies have improved quite a lot, but still they're costly, they're time-consuming. And as a result of that, to get a comprehensive ground truth network, you typically still have to rely on biological systems that may not match the exact problem that you're trying to solve, an exact system from which you have, for example, single-cell RNA-seq data. And generally speaking, this type of ground truth data is less available. And for a new context, it may be challenging to have. So a third option that has been used from early days of this type of problem is synthetic data. And for whoever has worked with method development, they're familiar with this idea of using simulation to test whether their method works well or not. And the challenge there a lot of times is that you can always, your insights that come out of it will rely on how well you have been able to simulate the data. And the quality of the simulation, if it has nothing to do with the real biology, well, your insights may not also have anything to do with real biology. So it becomes quite important that when we talk about synthetic data, we have high quality simulators that enable us to satisfy the constraints that we have. So in the context of single-cell RNA-seq data, there has been various powerful simulators developed already. For example, SCGAN is an example of that. SCDesign 2 and SCDesign 3 are other examples of that, where here, depending on the model they use, for example, the first one uses an adversarial approach. The second type of models rely on statistical properties of the data. These all enable us to simulate single-cell data that mimic the real reference data sets. However, they typically do not allow us to impose a specific gene regulatory network in the data. So what does that mean if our goal is to use our simulator for benchmarking different methods? Well, the implication is that we have more data, more simulated data, but we still don't know the ground truth that underlies that, right? And again, we need to have a way now for the simulated data to come up with a GRN that we can use for example, benchmarking. Now, that's one challenge, but the other challenge is that sometimes these models that do not try to explicitly impose any constraints, like kind of gene regulatory network or correlation or kind of interaction constraints, they may miss some of the interactions that actually exist in the data. So for example, I will show you today, for example, SCGAN fails to capture some of the relationship that exists in the reference data set. Now, there has been methods that have developed that allow you to impose a known gene regulatory network in your simulated data. For example, there was a famous study, Beeline, in 2020, where the authors benchmarked different GRN inference methods based on single-cell data. And in their benchmarking, they used both experimental benchmarks and also synthetic data that was generated using a method called Boole ODE, which uses differential equations in order to simulate data. Now, the challenge there was that the insights obtained from their experimental benchmarking did not match the insights coming from the simulated data. And in fact, some of the best performing methods on the simulation turned out to be some of the worst performing methods on the real data set, which again goes back to this challenge of whether our simulation actually gives us the insights that matches the real biology. Now, there are other issues with these models as well. For example, another powerful simulator, Sergio, it again relies on differential equations, but it also, to match the data to the reference data set and make the data look very realistic and have the properties of single-cell data, requires an iterative approach and a labor-intensive approach to generate such data. So the idea there is that, you have differential equations that allow you to generate clean data sets. But then if you want to make the data set actually have the statistical properties of your reference data set, for example, they allow you to add technical noise by changing 3 parameters. And they suggest that you check 5 different statistics and keep modifying these 3 parameters until you end up satisfied with the quality of your data, which again also adds the issue of subjectivity of, okay, when the user stops changing these parameters, well, is that good enough? Is that similar enough or not? So some degree of subjectivity comes in. But maybe more importantly, when we are using post-hoc distribution matching in this kind of with the user involvement, there's no guarantee that we are not actually affecting the causal GRN that we were intending to impose. And some of the interactions may no longer exist, and some new interactions we may have introduced. And that may explain why the authors observed that when they did this kind of iterative approach, the GRN inference methods that were performing well on their so-called clean data dropped to close to random after adding this kind of iterative process. Now, there are other issues. For example, these methods, because they are rule-based methods, they inevitably need to assume simplifying assumptions on how different transcription factors influence a target gene, for example, maybe an additive effect, and also do not readily preserve the gene and TF identities. So to address these and other problems, we developed GRouNdGAN, which is a robust causal generative adversarial model based on the CausalGAN framework that Yazdan described earlier. And this allows us to generate realistic single-cell data sets at both steady state and transient states. It allows us to capture the biological and technical noise properties of a reference data set automatically without this kind of iterative user manipulation. And a user-defined causal GRN is inputted to the model and is imposed in the data, and that can be considered the ground truth GRN that then we can use for GRN inference benchmarking. And also the other property that it has is that the relationship and the function and how different TFs regulate a gene in the data learn directly from the data itself through the neural network, which allows us to capture complex nonlinear transcription factor gene relationships. And finally, the gene TF identities are also preserved in the simulated data. So let's look at the architecture. And the architecture resembles and is basically based on the CausalGAN architecture. So we have seen this before, but let's just remind ourselves how the model and particularly GRouNdGAN works. So the GRouNdGAN training and model training follows a two-step procedure. In the first step, which I'm showing here, we simply train a Wasserstein GAN using a referenced single-cell RNA-seq dataset. So this is experimental dataset that we have. We have cells as rows, genes as columns. And the reason there is different color is that the shades of orange and red correspond to, for example, target genes. And here the columns that are blue or teal, they correspond to transcription factors or so-called regulators that we are considering. So the whole data set goes in. Here we have the typical Wasserstein GAN, where we have a critic, we have a generator, and as input, we provide it with a noise vector here at the beginning, and also a reference data set that I talked about, but no gene regulatory network is provided as input. Yes, question? Exactly. So that's the only thing we do. So one thing that happens here is that this data set, the reference data set, is library size normalized. And then we also have a library size or LSN layer here in the Wasserstein GAN that basically performs the same operation on the synthetic data. But no, it's not log transform. Yeah. OK, so here we have, again, two modules. We have our critic, which effectively, as Yazdan mentioned, is the discriminator, except that now it scores how real the samples look like. And during the training, we maximize the difference between the score that it assigns to X, which is our reference data here, and the score here that it assigns to the output of the generator, right? So we try to maximize this difference such that it's a good critic. It can tell the difference between simulated data and real data. On the other hand, generator, which plays an adversarial role with that critic, tries to generate vectors corresponding to single-cell RNA-seq data, but during the training, we maximize, so the weights of it are changed such that the score that the critic assigns to the output of it is very high, meaning that a very good critic thinks that its output are real. So this is the idea of the first step. But what are we using this for? Here, as I mentioned, after this is done, we are able to generate realistically looking single-cell RNA-seq data, but we have not imposed any type of causality in the model. So what we do is that now the generator of the model is taken from this network. The critic is completely discarded. And this generator is then used as the causal controller in the actual architecture of the GRouNdGAN. And not only that, but also we discard all the outputs of this generator corresponding to the target genes. So the only role that this causal controller plays for us is to generate the expression of the regulators of our model. And this is because we want to also impose an input gene regulatory network in the model. Okay, so here now we have our causal controller. It's already trained. Its weights are fixed. A noise goes into it. As its output that we end up using, it generates realistic transcription factor expression values. And a noise vector is also provided where these two go into the second set of module of the network here in the middle, where at least conceptually, for each gene, we have a different target generator that generates this expression. Now, how is that regulated, that gene? Well, as input, we also provided a gene regulatory network of this form. And we're going to talk about where that should come from or that can come from. But given this network, then the architecture and the connectivity of the output of the causal controller and the input of the target generators are guided by the connectivity of the GRN. So for example, here, in this GRN, gene 1 is regulated by only transcription factor 1. As a result of that, here as input, this target generator only accepts a noise value and the expression of transcription factor 1. For example, here for gene 3, it gets input from TF1, TFN, and a noise value, because here G3 is regulated by TFN and also TF1. Okay, so after these connectivities are connected this way, the output of this, which are the expression of the genes, are going to be concatenated with this kind of transcription factor expressions that are the output of the causal controller, and they're pulled together and again go into a library size normalization layer, LSN layer. And in the third set of steps or modules, again, we have the 3 neural networks. One of them is the critic, which is a Wasserstein critic, uses Wasserstein distance to score the realness of the samples. We know what it does. And then we also have the labeler and anti-labeler. So here, Yazdan talked about this, but I'm going to review that and also mention some differences between what we have and what the original causal GAN ends up doing. So in the case of the anti-labeler, as input, it only receives the generated or the simulated expression of the target genes, so only the cells that are in orange or yellowish kind of colors, not the blue ones. And then using those, it tries to estimate the output of the causal controller, right? So if you look at from this point all the way to this output, this area, it almost looks like an autoencoder idea, where you get the transcription factor values as input, and then you try to estimate the transcription factor values as output. Why do we do that? Because we want to constrain these generators and the latent space to ensure that these generators actually end up not only relying on noise, but also take advantage of the information that are in the transcription factors to really impose that causal connectivity that we talked about in the GRN. Now, we also have a labeler that does a very similar job, but in addition to accepting simulated target gene expression values to estimate, again, simulated TF expression values, it also accepts experimental expression values from here to estimate the actual experimental TF expression values. Now, Yazdan talked about the use of anti-labeler in the causal GAN to avoid condition mode collapse. But this is something that actually we didn't face in our problem. So we don't treat these as, we don't use a different mechanism for training them. So we, in the original model, The authors first maximize the loss for anti-labeler, then minimize it. We don't do that. We just minimize the loss both for labeler and anti-labeler in a similar manner. And what effectively it does is that while it may look like we are doing something redundant, the difference becomes that the anti-labeler makes sure that the simulated data is really imposing the interactions. The labeler, in addition to that, makes sure that the information also comes from the reference data set to make further connection to the real biology and real biological data. And I have a table with ablation study that shows that having both of them together improves the performance compared to having any of them, but you still can get a reasonable performance with just only, for example, having one of them. So now with this architecture in mind, how well does the model work? How realistic the models look like? So let's start with just some visualization. Well, first we trained our model, our simulator, based on human PBMC dataset corresponding to CD8 plus T cells from 10x Genomics. So we had approximately 21,000 cells here. This is a rather homogeneous dataset. And we had a validation set of 1,000 cells, a testing set of 1,000 cells, and the rest were used for training. And now we talked about the GRN needs to be inputted to data. So where does the GRN comes from? And this is something that we will talk more about later. But right now, just keep in mind that we use GRN-boost2, which is a well-respected method for GRN inference method on the training reference data set to find a kind of a scaffold GRN. And then from that, for each gene, we select the top 15 transcription factors and make the connectivity of the GRN through that. And that's what we impose for the figure that I'm showing. But more about this in a few slides. So from the t-SNE plot, here we can see, so here the red cells correspond to simulated cells. The blue ones correspond to real cells. And at least visually on the training set, we see that there is a good coverage of all the regions. There's a good mixing of red and blue. And similarly on the test set, which is what we really care about, we see a similar behavior, that there is a good mixing. Of course, this is just a visualization, and we want to really quantify the similarity. So we focused on 5 metrics. The first 3, cosine distance, Euclidean distance, and MMD, they correspond to the actual expression profile that we obtained. So it's not in a kind of a low dimensional, 2-dimensional or anything. And for these 3, we are comparing the simulated data and the reference experimental data for the test set. And we want this value to be as small as possible. Here, we also have a random forest, and it's the AUROC of random forest that tries to differentiate between simulated and reference data, and we want that value to be close to 0.5. And finally, we have here mean ILC value. And ILC effectively is a value that for our application, if it's close to 2 for a cell, it means that it's in its neighborhood, there is cells of both simulated and experimental. And if it's close to 1, it means that it is surrounded by only one cell type, one type of either simulated or real, which means that we want it to be as close as 2 possible. And here you can see the basically heat map on the t-SNE plot of how different cells will look like with regards to their ILC value. And we want this value to be as close as 2 to possible. So we have 3 benchmarks. These are all methods that automatically allow you to generate single-cell RNA-seq data without imposing any GRN. So one can argue that they may have an easier job because they are not having this constraint, this strict constraint of a causal GRN being imposed. And also, for a control, here we divided a reference experimental test data into 2 halves and calculated all these metrics on 2 halves of the real data to give us kind of a bound of what can be achieved if we were just looking at the reference data itself. And for all these measures, we are outperforming these alternative models, of course, except for the control, which is the bound. And this was encouraging. And then we wanted to see that, OK, what happens if we don't have this homogeneous data set for training? Because a lot of times in single-cell data, we deal with very heterogeneous data sets corresponding to many different cell types. And It is always possible, if we have information about what cell types are present to the data, to pass that on to a model. But it's not always an easy task to annotate all the different cell types. And we wanted to see whether our model is capable of generating realistic single cell data without having access to that kind of cell type or cell cluster information, or would we see mode collapse? And so without providing that cell type information explicitly, we trained a model on this data set that has approximately 70,000 PBMC from 11 different cell types. And here, again, visually, you can see the t-SNE plots for the training and the test. And you can see even in these kind of islands, we have a good mixing between the simulated and real data sets. And we can, again, look at the metrics and quantify this. So here I am comparing against the models that I just mentioned earlier, where none of them are provided cell type information at this point. Again, we see that in all 5 metrics, we outperform them. Then we also wanted to see how do we compare against these models or variations of them when they do receive cell type information. So here, for example, here we have conditional SCGAN, we have conditional WGAN, we have SC design 2 and sparsing where this kind of cell type information is provided. And when we have this full matrix, for 2 of the metrics, we are the best performing model. For 2 other ones, we are the second best performing model. And for 1 of them, we are the 3rd best. So overall, even though we are not in this experiment, we are not using the cell type information, we are performing quite comparable to models that are using this kind of cell type information. Now, the main question here for us was that, okay, we made realistic single cell data, but did we actually impose the causal GRN or not? Because that was really the main motivation here. So how do we test causality? Well, one way to define and maybe test causality is by interventional experiments, right? So for example, we may, if we have a GRN and we are able to perform some sort of knockout experiment on that particular transcription factor, then we can see whether it's the target genes, the expression of the target genes change significantly or not. And we can do this also using GRouNdGAN on the simulated data, because as Yazdan mentioned, this model allows us to sample from interventional distributions and not just observational. So the way we can do this is by, in the output of the causal controller for a particular transcription factor of interest, we can set its value to 0, which effectively does the kind of in-silico TF knockout experiment for us. And then we can do a forward pass while keeping other parameters fixed to see how the expression profile has changed. And we did that 1 TF at a time for all the TFs in our GRN. And for the genes, the target genes that were not targets of the knocked out TF, we did not see any change in their expression as expected. But the ones that were targets of that knocked out TF, their expression changed. So to quantify their change in their expression, we perform statistical tests. And here you can see the distribution of an adjusted p-value for this kind of change in the expression value of the targets of the knocked out TFs. And I should also mention that while the majority of them have a significant change in their expression, we cannot expect all of them to be very significantly changing, because we should remember that here we are performing 1 TF at a time knockout, but in our model, in our GRN, each gene is regulated by multiple TFs. So we cannot really expect that a knockout of a single TF would necessarily influence the target gene significantly, as other TFs may be more important for that particular experiment. Okay, so the next question we wanted to ask was that, okay, we have a model, we see that it generates realistic data, it imposes the causal GRN, but is this causal GRN such that a GRN inference method is capable of capturing it? And also, what happens to the edges that were important in the original reference data set, but we may have either failed to impose them or we may have decided not to impose them. So to test and answer those questions, we performed this kind of controlled experiment where we first used GRN-boost2 to find top 10 TFs for each gene from the reference data. Then we imposed, for each gene, we imposed the edge between that target gene and the TFs ranked 1, 3, 5, 7, and 9, and did not impose interactions between TFs ranked 2, 4, 6, 8, and 10. And this allows us to have kind of two GRNs of similar density, similar importance in the original reference data set, but we're imposing one and we're not imposing the other one, being able to use it as kind of a control to see how the behavior would look like. So if we start first by looking at this green line here, this shows the curve of the precision recall for GRN-boost2 now applied to the reference data set, experimental data set evaluated on this kind of positive control GRN that I mentioned. And here again, on the negative control GRN. And again, we see that very similar behavior and precision recall, showing that these two networks were of similar importance in the original reference data set for this experiment. But then when we look at GRN-boost2 applied to the simulated data, we see that we get a much higher AUPRC, showcasing that the edges that we actually imposed their importance in the simulated data have been increased. But very importantly, if we look at the negative control, we see that the importance of the edges that we did not impose actually has gone down compared to what they were in the reference data set, which is actually good news for us because it means that we are now disrupting those interactions that may have existed in the reference data set. Because if we want to now use this for benchmarking different GRN inference methods, we don't want interactions that exist in the reference data set, but we didn't impose to cause some sort of bias or error in insights we get from our benchmarking. Now, another thing that we should mention is that if we look at this curve, this is data generated by SCGAN, which doesn't explicitly impose the GRN. And when you compare this with the green dotted line, we see that the performance is now lower, which reflects that some of the interactions that existed in the original reference data set were not captured by SCGAN, which simply generates single cell data using a Wasserstein-GAN framework. Now, you may ask that, okay, is there anything unique about this imposed GRN? Maybe because you had the top TF in there, that is biasing your results. So to test that, we then switch the role of the imposed and the negative and positive. We imposed genes TFs rank 2, 4, 6, 8, 10, did not impose 1, 3, 5, 7, 9. And again, a very similar behavior was observed with regards to this kind of GRN inference benchmarking. Now, this allows us to answer three important questions. First of all, we saw that we can reconstruct the GRNs and the edges that we imposed in the simulated data. We saw that with GRN-boost2, we applied also other methods for GRN inference that have been in independent studies shown to perform well. And we see that they can also find these edges, of course, with different degrees of performance. We also saw that the non-imposed edges, but present in the reference data set, they did not still rank highly in the synthetic data. And finally, we also saw that interaction edges that preserved by the models that do not explicitly, and we did not, but preserved by the model, sorry. We also saw that interaction edges were not preserved by the models that do not explicitly impose them, such as SCGAN, at least to the extent that we have them in the reference data set. So one question that I said that I'm going to come back to this is this idea of using a GRN inference method on the reference data set to get a kind of scaffold GRN and then impose that. Is that a good idea? Does it even make sense? And why is that even different than, say, let's learn a GRN from the reference data and then use that and the reference data for the benchmarking? How is that different than this whole process of simulating data with a causal GRN? So the answer is that if we simply learn a GRN from the reference data and treat that as the ground truth, first of all, the edges that we find may not be causal edges. They may have a lot of spurious correlations. In fact, most GRN inference methods are not causal inference methods. And They may find simple correlations. And treating that as the ground truth will not be, it will basically lead to a ground truth that is very erroneous. Also, such a method will assume that a specific GRN inference method is superior than the others a priori because we are treating its output as the ground truth. And this is different from our approach because even if the GRN that we are finding from the reference data set is not causal itself, when we impose it using GRouNdGAN and generate simulated data, it turns into the causal graph of the simulated data, even if it was not the causal graph of the reference data. And in fact, we show that correlation-based methods, completely correlation-based methods, can also be used to find that kind of initial graph and then impose it in a causal manner using GRouNdGAN. The other important factor is that we don't have to impose the whole GRN that we are finding in the data. As we saw in the kind of controlled experiment we did, a very good practice is to keep some of the edges that were present in the original reference data set as a control and only impose a subset of the edges to then look at the difference between the behavior of the models on these. And this is the approach that, for example, we did and we recommend. Now, the other question that is kind of a follow-up of that is that does the GRN inference method that we use originally to find that kind of input GRN need to be a causal GRN inference method? And the answer is no. GRN-boost2 is not a causal inference method. We also use, for example, PP-core, which is a correlation-based approach. And again, a similar type of behavior can be observed when we find the initial GRN using PP-core and then try to reconstruct it using GRN-boost2 on the simulated data where we imposed, and on simulated data and the same data when we did not evaluate it on the edges that we did not impose. Okay. Now, another thing that we were interested in was the ability of the simulated model to capture the trajectories and the kind of continuous states that may be present in the data. Because a lot of times when we talk about single cell data, we are interested in its ability to capture trajectories, pseudo-time, and so on. So this was quite important for us. To test that, we applied our model to data corresponding to most mouse bone marrow, differentiating into different lineages, and then used PAGA, which is a trajectory inference method, to both using the simulated data and also experimental data, find the trajectories that are present in the test data set. And here, then, we used the markers of the cell types present in the data and their behavior of their activation to see whether we see concordance between these two. So here, for example, you can see the markers, the two markers of erythroid cells, that they're active in this region of the simulated data, and they're also active in this region of the experimental trajectory graph, which correspond to this region from the simulated data. Now, if we look at another set of markers for neutrophils, they are present in this region of the graph, while they're active in, again, the other end of the trajectory from the experimental data. And we repeated that process for all the markers of the different cell types present in the data, and there was a very high degree of concordance, and it allowed us to easily map these two trajectory graphs together. Now, after that, we wanted to see whether the pseudo-time ordering is also preserved, and to test that, we used a method called diffusion pseudo-time, and we found the pseudo-time ordering of both the experimental data here on the left and also simulated data. And then here, this last line shows the geodesic distance from some origin cell, from stem cells. So as we move away from it, we wanted to see how the markers of different paths that we identified in the data would change. So we identified 3 paths: erythrocyte, neutrophils, and monocytes. And then we looked at their markers. And on the left, you can see the behavior of the markers on the experimental data. And on the right, you can see them on the simulated data. And again, you can see a very high degree of behavior in how these different markers are getting activated in the pseudotemporal axis. And again, similarly for the markers of neutrophils and also markers of monocytes. So this was encouraging to us because, and it's important to us because a lot of GRN inference method, even if we are only focused on GRN inference benchmarking, rely on the idea of pseudo-time for GRN inference. Now, going back to one of our major motivations that we had, GRN inference benchmarking, we used this setup of positive control, negative control GRN, and we wanted to evaluate different GRN inference methods that were originally benchmarked in the Beeline study. As I mentioned, Beeline study performed benchmarking both on the experimental ground truth and also synthetic ground truth. And there were the results and the insights were not very consistent. So we used 5 GRN inference methods from the Beeline study, and using both the bone marrow dataset I mentioned and the PBMC dataset that I mentioned, with the setup that I described earlier, we benchmarked them. And here on the left, you can see their precision recall curve on the imposed GRN and the precision recall curve on the right on the unimposed edges, yet important in the original reference data set. So here you can see that, again, most of the models perform close to random for the unimposed edges, which is good, which is what we want to see. But we can see that there is a wide range of performances when we look at the imposed edges. And this was actually, there was a striking resemblance between the ordering we got from this benchmarking on the imposed edges and the ordering these methods had in the Beeline study on the experimental data and not on the synthetic data. In fact, the only difference between the ordering was that the order of these two methods were swapped in our ranking compared to their ranking. And this was, again, quite encouraging because, again, was another evidence that we are able to kind of bridge the gap between this synthetic data and real data. Now, one thing that I alluded to earlier, and we use this to check the causality imposition of the model, was this ability of GRouNdGAN in sampling not only from observational data, but also interventional data, which allows us to perform kind of TF knockout experiments. And this is something that we are quite interested in, and this is something that we are actively working to further improve and expand on. But as a proof of concept use case, we asked the question that how would the knockout, the in-silico knockout of top cell type specific transcription factors influence the generation of synthetic cells by GRouNdGAN? So if you think about cell type specific genes or transcription factors, a common way that we approach that is using differential expression analysis. So we compare a cell type against other cell types, find top differential expressed genes or TFs. And here, using the experimental data, we found top 3 TFs differentially expressed for the cell types. And then we knocked them out in our GRouNdGAN model. And then we looked to see how the cells are now being generated and comparing that before and after knockout. And one thing to remember, and one of the things that we are quite interested about GRouNdGAN, and reasons we are interested in it, is because it allows us to have matched before and after, matched wild type and knockout experiments for the same set of cells. So this can be quite powerful in kind of this type of analysis. Now, we generated data using the data set that had 11 cell types in it. Here I'm showing the Tisney plot, sorry, the UMAP plot, as well as the ILC distribution of the cells. We see a large MILC value, 1.94, showing that both simulated data and real data are mixing well. And here, and this is all before any knockout. And here, this is real cell density plot. And here, simulated cell density plot. Again, we see that these regions are all being generated, these islands. And then here, I'm showing you the results of knocking out the top 3 differential expressed TFs of CD19 plus B cells in this data set. So on the left, you can see the experimental reference data set. Blue are the cell type of interest. You can see most of them are in this island. There are some of them in this big region. And here is the simulated data after the knockout. And already we can see that this island, less cells in this island is being generated. And when we superimpose them on top of each other, we can further see that there is this region that is no longer being generated, corresponding to CD19 plus B cells. We can actually quantify this and look at different cell types. And here for example, I'm showing you the box plots, where on the left, the blue ones show the ILC value of a particular cell type, showing that they are relatively high, close to 2, and the same set of cells after the knockout of their top 3 differential expression TFs, and you see that less of the ILC value goes down, which shows that less of that cell is being generated by the model. So definitely there is some biological signal being captured here with regards to the relationship that exists between different cell types and the real biology. And this is something that we are quite interested in exploring further to turn the model into a predictor of TF knockout experiments. Now, one last thing that I wanted to mention is this idea of the role that each of these auxiliary networks play. So here is an ablation study table where we showing the 5 similarity metric as well as a value corresponding to how well the GRN was imposed. And here we are showing the percentage that the performance gets worse if we don't have a particular component. So if it's positive, it means that it's worse. If it's negative, it means that it actually gets better. And for example, if we don't have the labeler, the performance across the board gets worse. If we don't have the anti-labeler, again, we have worse performance. If we don't have any of them, much worse performance we get. And we can also even remove the noise. Technically, we can remove the noise here and just give TFs as input. And that also, particularly in the context of making the model, making the output looks like single cell, the performance becomes worse, even though it doesn't have a significant effect on the GRN imposition. And finally, the LSN layer that we talked about, it has a mixed effect. In some areas, it performs well, for example, for imposition of the GRN. But when it comes to similarity, for some metrics, the performance is better with it. Sometimes it's worse with it. And however, in spite of that, when it comes down to training and the stability of the model, it turns out to be a very important component to allow us to actually smoothly train our model. So in summary, I showed you GRouNdGAN that allows us to generate realistic single-cell RNA-seq data automatically. And within that, causal complex nonlinear TF-gene relationships are imposed. It preserves gene TF identities, trajectories, and pseudo-time orderings. It can be used for GRN inference benchmarking. And we are working on making this into an in-silico knockout experiment predictor as we go forward. So if you're interested in much more details, please check out the preprint version of the paper. Here is the QR code if you want to use that to reach the paper. And the code is available on GitHub, and there's a detailed tutorial of how to install it, for example, using Docker or local installation that you can, again, use the QR code or go to the GitHub. So finally, I wanted to acknowledge different trainees in my lab, especially Yazdan, who, along with Abdurrahman, led this project, and also acknowledge different funders that have supported this and other projects in my lab.